Hello, biology students. As promised, we're going to learn about proteins today and also enzymes, which are types of proteins. The lab that you set up yesterday actually is is about digestion, use of those enzymes, and also acid. So we're going to talk about that a little bit. Um, but proteins is the last biological molecule in this unit, so we're actually almost done with Unit 2. We've talked about carbohydrates, and we said carbohydrates are like sugars and starches, grains, all of those kinds of things. We've talked about lipids, and we've talked about nucleic acids. And so there's the protein is the fourth and final biological molecule that we're going to talk about. All right, so you are made of mostly protein. So proteins are pretty important in our bodies and in most living things, but especially in animals. We said that each of the four biological molecules has a monomer. And for proteins, that monomer are these amino acids. So again, I always think of the monomer as like one single little bead by itself, one single little structure. And then all of these amino acids link together to form larger molecules. So we have 20 different amino acids that are all linked together by something called peptide bonds. So peptide bonds are the bonds that hold these amino acids together. 20 different amino acids build all the proteins known to all living things on this planet. Okay, so there's 20 different ones. And think of it again as a bead. Like if you have even, let's say that Halloween's coming up, we have orange beads and we have black beads. The order that you can make a lot of different patterns with even just those two colors of beads. I could make a necklace that's orange, black, orange, black, orange, black, or I could do groups of 3 orange and 1 black and 3 orange and 1 black and so forth. So there's a lot of different combinations and patterns we can make out of those beads of amino acid. Now imagine if you had 20 different colors of beads, all the possibilities that there are. So in our bodies, our muscle tissues are predominantly protein. Our hair is protein. Our skin has proteins in. Our fingernails are a type of protein, but they're very different, right? And so the order that these amino acids are put together in ultimately determine that final shape of the protein. So I think of it like we have one like this, one like this, and so forth. So that's a simple polypeptide chain, maybe, and then these proteins fold up into these very complex structures. So here's what's important about knowing a little bit about those 20 amino acids. So if you've ever taken like drank a protein shake or taken a protein supplement or eaten a protein bar, a lot of times they will say on them, contains 20 different amino acids. Well, that just means that you're eating a protein. You're eating something that has protein in, and proteins, complete proteins, contain all 20 of those amino acids. So they're just careful to let you know that it contains all 20 amino acids. And the reality is you need all 20 of those amino acids in your diet. And most of us know things that are high in protein, and I guess I have it on here: meat, milk, eggs, cheese, beans, which are in the legume family, also tend to be high in protein. But if you're eating meat, you are consuming complete proteins and all of those amino acids already. Okay, our bodies can't make amino acids, so we have to eat them. We have to consume them. So it's really important to eat a diet that contains all 20 amino acids. So one of the things that people who don't eat meat, like some people just choose to not eat meat, they have to make sure that they're getting all 20 of those amino acids from plant sources, because if I'm not eating meat, I can't be assured, without a little research and being really careful, I can't be assured that I actually am getting all of those amino acids. And we need them in our bodies. Remember, I said we're made of mostly protein, so it's incredibly important that we get all 20 of those amino acids. So we have to consume them. And if we're getting them from plant sources, we have to do the research to make sure that we're getting all 20 of them from plants. All right. So proteins have, again, have a lot of functions in our bodies. One of the things that is just incredibly important to living things are proteins that build these very special structures called enzymes. So enzymes are catalysts. I don't know if you talked about that last year at all. So catalysts are things that speed up chemical reactions. And a lot of times they're reactions that would happen naturally, but they just happen like way faster with catalysts. And we see this all the time in chemistry, but we also see this in biology and biological molecules, in biochemistry, which is what this unit is about. So enzymes speed up chemical reactions, like breaking down your food, for example. So your food might break down eventually without enzymes, but it literally would not be fast enough to keep you alive. So we're very dependent on those enzymes. Enzymes are also important for chemical reactions that have to occur in our cells. And we have lots and lots and lots of different types of enzymes in our bodies, and they're all very necessary. Some diseases are caused by enzymes that weren't built correctly or weren't built at all, and then things go wrong, and we can't break things down fast enough, and sometimes maybe we make some enzymes but not enough of a particular enzyme, and that's really a problem for living things. All right, so these very specific proteins have a very specific shape. Each enzyme type has a job, and only one job, and it is to break down a molecule. Each enzyme is substrate specific. Okay, so each enzyme fits only one substrate. Okay, like a lock and a key that fit together, and you can't open the lock with a different key. It usually doesn't work. And different keys don't open different locks either. It has to be very specific, and it has to have that exact fit. So you can see my little picture here. The enzyme has a very specific shape and the active site is where that substrate, that molecule, maybe it's a food molecule, maybe it's something else, that is going to match that substrate shape. It's going to bond, and immediately, as soon as it bonds there, it breaks this molecule, and as soon as it breaks or cleaves or cuts this molecule, then it's released. And then this enzyme, guess what? It's just going to go find another one of these guys, break it down, release it. This enzyme can be used over and over and over and over again until it wears out. Okay, just like a lock and a key. Sometimes, and I see this with my lab stations in this room, sometimes if those keys are 15 years old, the keys that I use to open cabinets and so forth, they do wear out over time. Just the friction that's there, and metal gets tarnished, whatever, but they do get used up and they do wear out. So our body is constantly making new enzymes, of course, to replace those because they're really, really necessary. So enzymes, we say, are substrate specific. They will only fit one type of substrate molecule. This substrate molecule can only be broken down by this particular enzyme. So in your bodies, if you're missing it, you're just out of luck. And again, that's really a problem. Depending upon what enzyme it is, it can be incredibly deadly. So the lab that we set up yesterday with with all that saliva and all that meat. So the meat is just really building kind of an experiment to show you how acids affect proteins, because meat is a protein. So that's what you're going to be looking at tomorrow during the lab, seeing if there's any difference between the one in acid and the one in water. And we'll be evaluating that and collecting data. So that, because we have a lot of acid in our stomach, that's what we're looking for, the effects on that protein. The saliva ones, we have an enzyme. Saliva has an enzyme called amylase. And what amylase does is it starts breaking down starches immediately as soon as starches come in contact with it. That's why we put starch in those test tubes, and then we added amylase or saliva. We added saliva to one and water to the other, and we're just going to measure and see if it's been broken down. And I'll go over this procedurally tomorrow, but the starch test tubes, we're going to test them for the presence of sugar, which there was not any sugar in them. We only put starch in them. So we're going to test them for the presence of sugar and also for the presence of starch, which is a really easy test. And again, I'll go through that tomorrow. But that's what we're looking for in this lab. That's what we will be testing for and collecting data on tomorrow. So hopefully that makes sense. Please feel free to email me if you have any questions. And I hope you're enjoying the snow today.